is regulation of heart activity. Mechanism of heart activity regulation consists of two parts, central and local. Central part include nervous regulation. This influence sympathetic and parasympathetic nervous system, and humoral regulation. Local part consists of nervous regulation, metasympathetic part, and humoral and myogenic. Myogenic regulation include Frank-Starling law, unwrap effect, and Bowditch phenomenon. Myogenic regulation in local part. Myogenic regulation is implemented at the level of the myocardium itself without external influences. Frank-Starling law, the law of the heart's inflow load, the force of heart contraction depends on the end-diastolic filling of the heart with blood. The more blood flows to the heart, the more the cardiomyocytes will stretch, the greater will be the strength on their contraction. The Frank-Starling mechanism is based on the initial arrangement of actin and myosin news filaments in sarcomeres. If the filaments are stretched, the number of breaches formed during contraction will be greater, therefore the force of contraction will increase. Frank experiments on frog heart has found the ventricular output increases with the increase of saline pressure, which stretches the ventricular cavity. Starling showed on isolated dog hearts that the more ventricles are stretched by blood during diastole, the more the reduction in the next systole. On picture you can see scheme. The scheme shows heart lung operated by Starling. The essence of the Frank-Starling law. As a result, the law of the heart, Frank-Starling law, or heterometric mechanism of regulation, was derived the force of myocardial contraction fibers depend on the end-diastolic length. From the heart of the law implies that increased filling of the heart with blood leads to increased force of heart contractions. Reducing of the force of myocardial contraction observed when stretched more than 25% of the initial length. Such stretching is absent in the healthy heart, only 20%. So this act means that the number of breach of actinomyosin is the maximal during strain of each sarcomere to 2.2 microns, and force of cardiac contraction will depend on the number of formed breach. Unwrap effects. The greater the pressure in the aorta, the greater the strength of heart contraction. The mechanism of implementation of these effects consists in improving myocardial trophicity, coronaries and supplied with blood direct from the aorta. The higher the pressure in the aorta, the more blood will circulate through the coronary arteries, that more nutrition they receive, the more they will contact. In addition, increased blood supply to the coronary arteries will lead to increased stretching of cardiomyocyte, and accordingly the strength of their contraction will increase. Bowditch phenomena. On scheme, you can see electrocardiograms during different conditions. An increase in heart rate leads to an increase in the force of heart contraction. The mechanism: an increased heart rate leads to a gradual accumulation of calcium ions in the myoplasm. In cardiomyocytes, it does not have time to be completely pumped into the tanks due to a decrease in diastole time. The more calcium ions, the more actin-myosin breach will be formed, the greater will be force of heart contraction. However, the force of contraction does not increase without limit cardiac output reach maximum values of heart rate near 150 per minute. With a further increase in heart rate, the force of contraction gradually decreases, because there are no more free active actin centers and myosin heads in sarcomeres. They are all involved in contraction. This addition is the cause of the instant death of athletes. The more heart rate, especially calcium ion, due to be lack to ATP, relaxation of the myocardium cannot develop, and contracture of the heart develops, and it stops in systole. Next, information about local mechanism of heart regulation with nervous part. In the heart, local cardiac reflexes are implemented, the arcs of which close at the level of intramural metasympathetic ganglia. Intracardiac reflexes are able to change the contractile activity of different parts of the heart. An example of such a reflex can be the fact that when the blood flow to the right atrium increases, the force of contraction of the left ventricle increases. The intracardial nervous system is normally in contact with the central and peripheral nervous system, and to some extent is subject to its influences. But after the transection of the extracardial nerves, the intracardial nervous system fully ensures the regulation of heart activity. The evidence is a change in cardiac activity during physical exertion in people with transplanted hearts. Next, information about local mechanism of heart regulation with humeral part. Recent studies have proven that the heart is an endocrine organ because cardiomyocytes can synthesize substances such as atrial natriuretic peptide, reduce the force of heart contraction, catecholamines, renin, digitalis-like substances, and others. Effect of atrial natriuretic peptide. On skin, you can see different influence natriuretic peptides or factors on vessels and urine system. Vasodilation. This fast effect on vessels, like result, decreased blood pressure. Second effect of natriuretic peptide is effect influences on kidney. Increased urine output, water elimination, decreased blood volume, and decreased blood pressure. This affect natriuretic peptides. Next table show effect catecholamines on heart activity. Different concentration catecholamines on plasma give different results. Cardiac effects change heart rate. Epinephrine and norepinephrine give effect increase heart rate. Inotropy effect on contractility give effect increase contraction muscles, epinephrine and norepinephrine. Dromotrophy effect, this effect on conductive system, increase conduction velocity, have result influence epinephrine and norepinephrine. The mechanism of catecholamines action on the heart. Catecholamines interacting with beta adrenoreceptors of heart cause activation of enzymes adenylate cyclases, which converts adenosine triphosphoric acid in cyclic adenosine monophosphorate. Increasing of intracellular concentration of cAMP cause activation of cAMP-dependent protein kinase, which catalyzes the phosphorylation of proteins, which leads to an increase in entrance of sodium and calcium into the cell. This effect you can see on scheme. Catecholamines have connection only with adrenergic receptors. And on scheme you can see reaction epinephrine and norepinephrine for connection with different type receptors. These present different type adrenergic receptors, alpha 1s, receptor present in vessels, beta 1 receptors present on heart, and beta 2 receptors, adrenergic, present on vessels, but vessels in skeletal muscles and liver. Effect connection adrenergic receptor with epinephrine and norepinephrine have difference. Connection with alpha 1 receptors, epinephrine and norepinephrine, give effect vasoconstriction. Epinephrine and norepinephrine have influence on beta 1 receptors. Like results, give positive effects. Increase heart rate, increase contractility, increase excitability, and increase conduction velocity. With beta 2 receptors have connection epinephrine only, and like results give opposite effect vasodilation. This effect present in skeletal muscles and liver. Next, information about action of acetylcholine on the activity of the heart. Mechanism activation you can see on these pictures. In the external membrane of cardiomyocytes, muscarinic cholinergic receptors are dominate, similarly as beta adrenoreceptors. The density of muscarinic receptors in the myocardium depends on the concentration of the antagonist. During interaction with muscarinic receptors, acetylcholine primarily cause inhibition of adenylate cyclase activity and secondary activates guanylate cyclase, increase of intracellular concentration of cGMP, causes activation of acetylcholine dependent potassium channels. It leads to increase of potassium ions out of the cardiomyocytes. Due to increased release of potassium occur hyperpolarization of cell membranes. Effect of change of extracellular concentration potassium ion on the heart activity. When the concentration of potassium ion equal from 4 to 8 millimole per liter is small depolarization, the membrane potential decreases from minus 90 to minus 80 millivolts. Condition of sodium ions channels of sarcolemma does not change significantly. Excitability of muscle fibers and the speed of pulses increases. This result you can see on start electrogram. When the concentration of potassium ions equal from 88 to 35 millimole per liter, change membrane potential. Membrane potential decreases from minus 80 millivolt to 40 millivolt. Significantly reduce conductance of fast potential-dependent sodium ions channels, relative refractory periods. The result is reduction of excitability and conductivity and changing of nature of the action potential. Reducing of the potassium extracellular concentration, hypokalemia, causing hyperpolarization of the membrane. If hypokalemia up to the level of 2.3 millimole per liter, the threshold depolarization significantly increase. The anxiety decrease. The duration of action potential and force of heart contraction increases. Effect of change in extracellular calcium concentration of the heart activity. In a solution without calcium ions, isolated heart stop quickly. The reasons for this is a complete rift between excitation and contraction. Normally, during phase plateau, calcium ions, which enter from the extracellular space into sarcoplasm of cardiomyocytes, start calcium ion release from the sarcoplasmic reticulum and replenish its reserve in this structure of muscle fibers. Reserve of calcium ions in the sarcoplasmic reticulum rapidly depleted during reducing of the extracellular concentration. Also, calcium ion concentration in the sarcoplasm decrease and thus reduce the strength of heart rate. Increasing of the calcium ions concentration in the plasma leads to reduction of myocardium excitability and contractility as an extreme expression of this positive inotropic action of calcium ions, cardiac arrest in systole arise. Reason is the binding of calcium ions with troponin that allow actin and myosin threads to interact that provide myocardial contraction. If calcium level decrease, the decrease of excitability and contractility of the heart observed. Next, information about effect of thyroid hormones of the heart. Thyroxine and triiodothyronine is hormones from thyroid glands. Like reactions have effect on heart activity. This effect same like effect catecholamines, because these hormones regulate isozyme composition myosin. These hormones increase the number of beta-adrenergic receptors and their sensitivity to catecholamines. These hormones reduce the number of muscarinic cholinereceptors. Results, maybe effect, influence on thyroid hormones have positive effects: increase heart rate, increase excitability, increase conductibility, and increase contractility. Effect of corticosteroids on the heart. Glucocorticoids, which produced in adrenal glands, have effect increase beta-adrenergic receptors and sensitivity receptors to catecholamines, because results influences have same like catecholamines positive effect: chronotropy, batmotropy, dromotropy, and inotropy effect. Next, information about reflexes from atria. When atrial pressure increased due to increasing blood volume, atria stretched. Signals pass to afferent arterioles in kidney to cause vasodilation and glomerular capillary pressure, thereby increasing glomerular filtration. Signals also pass to hypothalamus to decrease antidiuretic hormone secretion and so fluid reabsorption. It causes decrease in both blood volume and arterial pressure to normal. Other reflex reaction is known as atria and pulmonary arteries reflex. When atria pressure increase due to increase in blood volume, atria stretched. Low pressure receptors, similar to baroreceptors in atria and pulmonary arteries, stretched and stimulated. Signal pass to vasomotor centers and inhibit vasculomotor area. arterial pressure decrease to normal. Next slide shows Bainbridge reflexes. Increase intravascular volume, your results stretch arterial receptors, reaction on medullary activation, and increase sympathetic activity to sinoatrial nodes. Direct stretching of SA nodes give effect increase heart rate. This information you can see on scheme connection heart with vasomotor centers. You have picture with Bainbridge reflex. On table, you can see reaction heart on intravenous infusion. Intravenous infusion give results arterial stretch and like effect, fast time decrease sympathetic activity to kidney and increase urine output. Second time, decrease vasopressin production, antidiuretic hormones from hypothalamus, and increase urine output and decrease water absorption and regulation of blood pressure. And last effect, increase concentration atrial natriuretic peptide or factors. Like results, increase effect natriuretic peptides on urine and decrease blood pressure. Next, information about location of receptor in the heart. Heart muscles contains both chemical and stretch receptors in coronary vessels. All heart, cameras, and pericardium. Stretch receptors are irritated by changing blood pressure in heart cameras and vessels. Chemosensitive cells, which are stimulated by decreased oxygen and increase of carbon dioxide and hydrogen ions, and biological active substances also are called as chemoreceptors of the heart. Reflex reaction from receptors of pericardium, endocardium, and coronary vessels. These reflexes regulates heart activity. Reflex reaction from receptors of pericardium, endocardium, and coronary vessels lead to stimulation vagus nerve. It leads to parasympathetic stimulation of the heart. Parasympathetic stimulation causes decrease in heart rate and contractility, causing blood flow to decrease. It's known as negative inotropic dromotropic, bathmotropic, and chronotropic effect. Central mechanism of heart activity regulation. On scheme, you can see effect influences different part of brain on the heart. Prefrontal cortex have effect resting higher frequency heart rate. Sympathetic activity block, and on electrocardiogram higher frequency heart rate velocity. Insular cortex have effect during atrial arrhythmias, cardiac hypertrophy, disruption in diurnal blood pressure, myocardial injury, breathing disorders during sleep, and ECG change like atrial fibrillation and depression ST and inverted T waves. Effect of hippocampus have during hypertension, myocardial infarction, atrial fibrillation, heart failure, and atrial fibrillation on electrocardiograms. Brainstem have irritation during ventricular arrhythmia, myocardial infarction, bradycardias, sudden death. On ECG, result excitation is ventricular arrhythmias. Effect of cerebral cortex on heart activity. Cortex is the organ of mental activity. It provides a holistic adaptive response. The work of the heart depends on the functional state of the cerebral cortex. Thus, in athletes observed pre-start condition manifesting with increasing of heart rate, hour passing, and anxiety state reduction of the can be achieved. The effect of stimulation of the cerebral cortex occurred during stimulation of motor and premotor zones, cingulate gyrus, orbital surface of the frontal lobe, the anterior region of the temporal lobe, usually an increase of heart rate observed here. Effect of hypothalamus of heart activity. The hypothalamus plays a central role by integrating the information conveyed via various signals input with the neural networks involved to determine and coordinate the appropriate responses to maintain homeostasis. Much of the investigation of the cardiovascular regulation by the hypothalamus has focused on the paraventricular, dorsomedial, lateral, and posterior nuclei. Consequently, there is wealth of information about the anatomical, cellular, and molecular processes underlying the control of autonomic and cardiovascular function by these hypothalamic nuclei. Thus, in hypothalamus are structures that regulate the activity of the heart. Often, but not always, electrical stimulation of the anterior hypothalamus leads to decreased cardiac and rear to increase heart rate. Connection hypothalamus with different part of the heart you can see on scheme. Control of heart activity by cardiovascular center. The medulla oblongata, specifically the medullary cardiovascular center, is the primary site of cardiovascular and baroreflex integration. In this region, the nucleus of the solitary tract served as the primary site for the first synapses of the baroreceptors afferents and is the key integrating site for all baroreceptors input, including the cardiopulmonary baroreceptors. Lateral portion of vasomotor center transmit excitatory signals through the sympathetic fibers to heart to increase its rate and contractility. Medial portion of vasomotor center transmit inhibitory signals through the parasympathetic vagal fibers to heart to decrease its rate and contractility. Neurons, which give impulses to the heart, have constant level to activity even at rest, which is characterized as nervous tone. Scheme movement impulses from cardiovascular centers you can see here. Effect through the medulla oblongata and spinal cord and through the different part of autonomic nervous system, vagus and parasympathetic and sympathetic influences. On scheme, you can see result influences cardiovascular centers, and tables show input of cardiovascular centers, nerve impulse from higher brain centers is cerebral cortex and limbic system and hypothalamus. From sensory receptors, this peripheral part, proprioceptors, motor movements, chemoreceptors, give result monitor blood chemistry, and baroreceptors result monitor blood pressure. All impulses move to cardiovascular centers. Like results, you can see effect cardiac acceleration through the nerve sympathetics. Output to heart. Increase frequency of nerve impulses. Give effect increase rate of spontaneous depolarization in SA node and AV nodes and increase heart rate. Next effect, increase contractility to atria and ventricle increases stroke volume. And last effect, maybe decrease rate of spontaneous depolarization in SA node and AV node. Give effect decrease heart rate. Last effect, show reaction parasympathetic part of autonomic nervous system. But remember, through the cardiovascular center. Peculiarities of the vagal innervation of the heart. In the medulla oblongata, the nucleus of vagus nerves located the axons of the cells of the nucleus within the right and left nerve trunks, sent directed to the heart. Right vagus nerve fibers are distributed mainly in the right atrium. The associated neurons innervate myocardium, coronary vessels, and sinoatrial node. As a result of the structural features of the right vagus nerve, stimulation of it shows its influence on heart rate. Left vagus nerve fibers transmit the effects to atrioventricular node. Green color lines show this information on scheme. As a result of the structural features of the left vagus nerve, stimulation of the shown is influence on atrioventricular conduction and contractility of cardiomyocytes. Have effect on heart rate and contraction. Parasympathetic stimulation causes decrease in heart rate and contractility, causing blood flow to decrease. Is negative inotropic effect, dromotropic effects, bathmotropic effect, and chronotropic effects. Influence of sympathetic nervous system activity of the heart. In structure, the fast neurons of the sympathetic nervous system that transmit impulses to the heart located in the lateral horns of the upper for the 5 thoracic segments of the spinal cord, from T1 to T5. Processes of these neurons terminate in the cervical and upper thoracic sympathetic nodes, where the later neurons process which go to the heart. On scheme, you can see movement impulses through the sympathetic tracts from cardiovascular zones through the lateral horns to heart. This picture shows structure sympathetic chains. From both sides you can see sympathetic paravertebralis ganglions, and sympathetic nerves, connects ganglion with heart. This you can see sympathetic nerves have connection with myocardial cells of the heart and short connection with SC node. Sympathetic effects. Sympathetic nerves from T1 to T5 control activity of the heart and large vessels. Fast neurons lays in lateral horns of spinal cord. Second neurons located in sympathetic ganglions, and sympathetic nerve system gives to the heart vasoconstrictor and vasodilator fibers. Vasoconstrictor impulses are transmitted through the alpha adrenoreceptors, which are more spread in major coronary vessels. Transmission impulses through the beta adrenergic receptors lead to dilation of small coronary vessels. Sympathetic influence produce positive inotropic effects, dromotropic effects, bathmotropic effects, and chronotropic effects. Give positive contractility, excitability, contractibility, and heart rate. Mechanism for difference part autonomic nervous system. First time for sympathetic and second time to parasympathetic. Heart rate. Sympathetic nervous system give effect increases after reaction with beta 1 adrenergic receptors. Parasympathetic effect decrease action with effect with muscarinic receptors. Conduction velocity. Sympathetic nervous system give effect increases, parasympathetic effect decreases. Contractility. Sympathetic nervous system give effect increase contractility after reaction with beta 1 adrenergic receptors, and parasympathetic give effect decrease atria only after reaction with muscarinic cholinergic receptors. Next, information about irritation of visceral receptors and effect on heart activity. Irritation of visceral receptors result in stimulation of vagal nuclei, which cause decrease in blood pressure and heartbeat. Parasympathetic stimulation causes decrease in heart rate and contractility, causing blood flow to decrease. It's known as negative inotropic, dromotropic, bathmotropic, and chronotropic effect. The mechanism is important for doctor is performing diagnostic procedure when probes from apparatus are attached into visceral organ. This may cause excessive irritation to visceral receptors. Next, information about reflex regulation of heart activity from extracardial receptors. Reflexes from aortic carotid zones. Reflexes effect from internal hollow organs. Reflexes effect from the trigeminal nerve, skeletal muscles, lung tissue. These reflexes from extracardial receptors for change heart activity. And first time information about baroreceptor reflexes. There are many receptors which send afferent fibers to the cardiovascular centers. Some of these receptors reside within the cardiovascular system, whereas others are located outside of the circulation. One such is the carotid baroreceptors, located in the wall of the carotid sinus, which stimulates the origin of the internal carotid arteries and the carotid bifurcation. The other set is aortic baroreceptors, located in the wall of the carotid sinus, which is situated at the origin of the internal carotid artery and the carotid bifurcation. The other set is the aortic baroreceptors located in the wall of the arch of the aorta. The afferent impulses from the carotid sinus travel in the glossopharyngeal nerves, whereas those from the aortic arch are conveyed by the vagus nerves. The baroreceptors are stretch receptors which are activated when the vessel's wall is stretched by the arterial pressure. These receptors are not only sensitive to the absolute pressure level, but also to the rate of change of arterial pressure. That the frequency of afferent impulses increases when the process pressure is rising during systole and decrease when the pressure is falling during diastole. Next information about regulation blood pressure with different conditions for effect from baroreceptors. In left side you can see different conditions with blood pressure increase and blood pressure decrease. Blood pressure increase. A sudden increase in blood pressure is detected by the baroreceptors in the internal carotid arteries and aorta, which affects the baroreceptors reflex. Next impulses go to cardioregulatory centers and increase parasympathetic stimulation of the heart and decrease sympathetic stimulation of the heart and adrenal medulla. Like results, decrease heart rate and decrease stroke volume, and autonomic nervous system stimulation of the heart, decrease heart rate and stroke volume result from the decreased release of epinephrine and norepinephrine from the adrenal medulla. Result reaction: the blood pressure decreases because of the decreased cardiac output in the heart, resulting from the decreased heart rate and stroke volume. Down part of the table show condition when blood pressure decreases. This you can see a sudden decrease in blood pressure is detected by the baroreceptor in the internal carotid arteries and aorta, and start baroreceptor reflex. Like result, impulses go to cardioregulatory centers, decrease parasympathetic stimulation of the heart, and increase sympathetic stimulation of the heart. Result stimulation increase heart rate and stroke volume through the release of epinephrine, higher concentration from adrenal medulla, and stimulation of autonomic nervous system, sympathetic part. Like results, to blood pressure increase because increased cardiac output resulting from the increased heart rate and stroke volume. Blood pressure homeostasis is maintained. On scheme, you can see connection between different parts of brain and medulla, of adrenal medulla and heart. This you can see parasympathetic and sympathetic nerve fiber connection with different part of the heart and different type reaction. This table shows reaction chemoreceptors on change blood pH. In level part, you can see reaction on blood pH increases. Blood pH increases. An increase in blood pH, often by a decrease of blood carbon dioxide, is detected by chemoreceptor in the medulla oblongata, which affects the chemoreceptor's reflexes. Next, impulses go to cardioregulatory centers and increase parasympathetic stimulation of the heart and decrease sympathetic stimulation of the heart and adrenal medulla. Like results, decrease heart rate through the decreased parasympathetic part and through the decreased concentration epinephrine, and decrease in blood pH result from decreased blood flow through the lungs. The decreased blood flow results from the decreased cardiac output caused by decreased heart rate and stroke volume. Down parts show reaction chemoreceptors on lower concentration of pH. Blood pH decrease. A decrease of blood pH give effect start regulation in medulla oblongata and start regulation chemoreceptor reflex. Impulses go to cardioregulatory centers. From centers, impulses go to heart and increase heart rate through the effect autonomic nervous system and higher concentration of epinephrine. Like results, increase blood pH through the increased blood flow through the lungs and increase cardiac output, heart rate and stroke volume. Reflex from proprio, thermo and interoreceptors. Contraction of skeletal muscles during exercise compress blood vessels into heart, increase cardiac output, and increase arterial pressure. Stimulation of thermoreceptors cause spreading impulses from somatic sensory neurons to autonomic nerve centers and so leads to change in tissue blood supply. Irritation of visceral receptors results in stimulation of vagal nuclei, which cause decreasing blood pressure and heartbeat. On table you can see cardiovascular adjustment to exercise, skeletal muscle activity, give effect, improved venous return, and metabolic vasodilation in muscles. Improved venous return give effect, increase stroke volume, and like results, increase cardiac output. Cardiac output give effect, increase blood flow through the skeletal muscles. This mechanism regulation blood flow with help cardiovascular system and skeletal muscles. Respiratory arrhythmia affect influence lungs reflexes on heart activity. The primary mechanism of respiratory sinus arrhythmia are understood to be modulation of cardiac vagal efferent activity by the central respiratory drive and the lung inflation reflex, and the degree of respiratory sinus arrhythmia increase with cardiac vagal activity. In case of respiratory arrhythmia, significant role belongs to impulses from lung receptors, mechanoreceptors of atrium, that's responsible of increase of flow of venous blood to the heart. On electrocardiogram, you have different heart rate during inspiration and during expiration. Next table shows effect of hypocarbia on cardiac activity. Hypocarbia, condition with higher concentration of carbon dioxide, have indirect and direct effects on heart activity. Indirect effects: sympathetic nervous system activation, will result increased heart rate, increased cardiac output, and increased contractility. Direct effects give effect depressed myocardial contractility. Different concentration carbon dioxide give different results on heart activity. Thank you for your attention.